Anthrax is an aerobic, infectious, and sometimes fatal disease in animals and humans caused by a gram-positive, non-motile, endospore-forming bacterium called Bacillus anthracis. It is a multi-species disease that can infect most of the taxonomic groups and can also be found naturally in soils throughout the world. The particles of anthrax that are infectious are the Bacillus anthracis endospores. The organism penetrates into the bloodstream and harms the host by producing toxins within the body. The slimy capsule layer that surrounds the organism allows it to resist phagocytosis by white cells. Ruminants and hindgut fermentators are the most susceptible, seeing that they are grazers, while carnivores and primates have a lower chance of infection.
Once ingested, the spores are activated and the bacteria begin to reproduce. Reproducing bacteria make the proteins that combine to form toxins. Bacillus anthracis has two components to its virulence: the toxin it secretes as well as its capsule. The capsule prevents post-phagocytosis when Bacillus anthracis is in the vegetative form. The toxin has three proteins: the edema factor, lethal factor, and protective antigen. Protective antigen is a binding domain that allows the toxins, edema, and lethal factors to enter host cells. Lethal factor causes immunosuppressive effects, damages endothelial cell function, and causes cell apoptosis. It also disrupts downstream signaling, which is important to normal cell functioning. Edema factor works additively with lethal factor and also has immunosuppressive effects. Edema factor causes swelling and fluid buildup in the body by triggering an adrenaline overload. The cells become hyperactive and devour the chemicals they store for energy. They lose the ability to regulate their environments and die, releasing water in surrounding tissues. The buildup of fluid, known as edema, around the lungs is fatal and can kill infected cells. Animals can no longer perform their normal bodily functions, therefore resulting in their rapid death.
We will now be discussing the most common portals of entry of anthrax. So anthrax has a few portals of infection, including cutaneous, inhalation, and via the GI tract. So before discussing this, it is important to know that anthrax spores that have been deposited into soils via blood from a contaminated carcass prefer alkaline soils rich in calcium and can survive in these favorable conditions for roughly 3 years. The calcium-rich soils form the calcium lattice that is important for the survival of the anthrax spores. So as mentioned, the anthrax has a few portals of entry. However, in wildlife, the portal of infection varies depending on the animal's feeding behavior and the animal's susceptibility to the bacteria. So carnivores are highly resistant to anthrax spores found in contaminated carcasses and need to require a large amount of these spores to become infected. However, due to their feeding habits, which include eating a large amount, it is possible for these animals to become infected. Herbivores, on the other hand, are more susceptible to anthrax, and the portal of entry of this bacteria can occur in multiple ways. However, the GI tract is the most common. Herbivores become infected through eating contaminated vegetation. It is suspected that herbivores grazing on fields that have rough vegetation that contain anthrax spores in the soil becomes infected via the GI route. The rough vegetation causes small cuts in the animal's GI tract, which causes an entry way for the anthrax into the GI tract. Anthrax can also be transmitted via a vector, such as blowflies. Herbivores can also become infected via inhalation of anthrax spores. This is a less common portal of infection, however, it does occur. In times of drought, followed by a heavy rain period, anthrax spores in soils get carried by the water to low-lying areas. Herbivores such as buffalo can disturb the soils in such depressions through feeding and mating behaviors, which causes clouds of dust filled with spores which the animal inhales. Water also plays a really big role in the transmission of anthrax due to scavengers such as vultures and hyenas feeding on an infected carcass and spreading the anthrax spores to water holes. Vultures, which do not get infected themselves, spread the anthrax spores through bathing and cleaning themselves after eating a contaminated carcass in a shared water hole. So now that we have looked at the portals of entry of anthrax into a host, we will look at the exit route the bacteria uses to exit the body and disperse and infect even more hosts. So anthrax basically survives through killing its host, and it is released into the environment primarily via blood and tissue from the infected animal. The vegetative form of anthrax is fragile and not resistant to unfavorable environmental conditions. Therefore, the bacteria forms spores in unfavorable conditions, which are more resistant. A host is infected through consuming or inhaling anthrax spores, and the favorable environmental conditions of the host body causes the anthrax spore to convert into a capsulated vegetative bacterium. Once anthrax has killed the animal, the oxygen levels within the host drops to such levels that the vegetative bacteria have to exit the body to survive and disperse further. As mentioned, it does this through blood and infected tissues. Anthrax is therefore very dependent on scavengers in humans to open the infected carcass and expose the blood and tissues to oxygen. Anthrax interferes with the animal's ability to form blood clots and therefore pools of blood are formed once the carcass has been opened by scavengers, and this also causes the animal to bleed through its natural orifices. The vegetative form of the anthrax bacteria converts into spores when it comes into contact with oxygen. It does this because the anthrax spores are more resistant to environmental conditions and therefore it's a more favorable state. So sporulation occurs once the carcass has been opened by scavengers and the tissues and blood are exposed to oxygen.
The following are pictures of blood smears being made in the field. Laboratory diagnosis of anthrax. In the laboratory, the main stains that are used are Giemsa or Macfadyen stains. A Macfadyen stain is a polychrome methylene blue stain, and it is the simplest, most reliable method for confirming the presence of capsulated B. anthracis. Cultures in the laboratory should be kept at the appropriate biosecurity levels. The confirmation of anthrax can be done by phage typing. A species-specific phage is used routinely in the identification of Bacillus anthracis, the causative agent of anthrax. Thus, reproduction of anthrax in laboratory animals is not necessary to confirm identification of this organism. Plate agar is the best culture medium. It is a selective media used for the isolation of Bacillus anthracis from contaminated specimens. Plate agar inhibits most contaminating organisms and spore formers closely related to the B. anthracis. The specimens are first heat or alcohol shocked, and then dilutions of these preparations are sprayed across the plates. Circular creamy white to gray-white colonies with ground glass texture are subcultured into appropriate media to test for gammaphage, penicillin susceptibility, lack of motility, and lack of beta hemolysis for complete identification of the B. anthracis. The Ascoli precipitin test is an old test developed in 1911 to supply rapid retrospective evidence of anthracis infection in animal products. It was designed to detect B. anthracis antigens in tissues of animals and their byproducts. The test makes use of commercially spread serum harvested from hyperimmunized rabbits, which react with the boiled filtrate of the suspected tissues in a test tube. A line of precipitation develops at the antigen-antibody interface. In this picture, we can see the culturing of anthrax on the plate agar. Here, we've got a buffalo blood smear with a low terminal bacteremia. Here, we can see the B. anthracis microorganism, but very few of them in this blood smear. In the next blood smear, we've got a kudu of a high terminal bacteremia. Here we can see multiple B. anthracis microorganisms in the blood smear.
Precautions when dealing with anthrax samples. Precautions should be taken by veterinarians and other personnel to avoid skin contact with potentially contaminated samples. Precautions include personal protective equipment, such as impermeable gloves, clothing, and boots. Disposable protective equipment should be used, but if there is none available, the used equipment should be completely decontaminated. The risk of respiratory infection is extremely low, but it is still recommended to wear respirators for safety. When shipping samples, the primary sample container should be placed within a secondary leak-proof container with absorbent material to absorb any liquid in the event of a spillage. Packs for cooling should also be placed inside the secondary container.
Disinfecting the contaminated environment. The management principles used in the control of anthrax in wildlife are the same as the principles used for livestock. It is important to note that anthrax is also part of some natural ecosystems. It is for these reasons that disinfecting the contaminated area is extremely difficult. The following are methods to be followed to disinfect a contaminated environment. Quarantine. Once an anthrax outbreak has been identified in a particular area, it is important to implement strict quarantine parameters to prevent the spread of bacteria. Carcass disposal. The intact carcass should be burned. Fire should be rapid and hot enough so as to prevent the spread of the bacterial spores. It is also important to burn surrounding infected soil and feces. The carcass can also be buried. However, scavengers could dig up the carcass, therefore spreading the bacterial spores in the environment. Scavenger control. Treating the carcass with formaldehyde will discourage scavengers from disturbing the carcass. Local site disinfection. Disinfection and decontamination of the anthrax outbreak site using chemicals like formaldehyde and chlorine solutions, as well as removing contaminated soil 20 centimeters deep. Treatment. Long-acting antibiotics should be used as a treatment. Vaccines. Darting wildlife with vaccines is very costly. However, oral vaccines are more cost-effective.